Hello everyone, my name is Chris Worley. I'm an entomologist with the USDA in Poplarville, and one of the specialty crops that we're promoting and doing some research on are the Muscadine grapes. And one problem that we're looking at is their susceptibility to Xylella, and I'll be talking today about some of the potential vectors of this bacterium, the leafhoppers. Xylella is a bacteria that can cause several different leaf scorch diseases depending on the crop and on the subspecies that's being vectored. Here you can see a picture of an olive orchard in Europe that's been decimated by Xylella. And in addition, there's an almond leaf scorch and oleander, but the most prominent one here in the States is in our grape industry with Pierce's disease. There are protocols available to identify the subspecies and determine the infection status of plant and insect samples. And so that's the direction that we were taking our research in. Leafhoppers are kind of a newer group to me. There's just fascinating diversity, over 3,000 species in North America, and a lot of them are really brilliantly colored, and I've really been enjoying looking at them on the microscope. But they also are, since they are xylem feeders, they are effective vectors of Xylella, and vector management has been an important strategy in limiting the spread of the disease in grape vineyards out west, and it may be a potential area to look at here in the southeast. In the southern U.S., there's multiple generations per year. They're active year-round. They're hard to control because they're so small, highly mobile, but we do have some good products available. The most important leafhopper in terms of disease vectoring is the glassy-winged sharpshooter, and it is native to the southeastern U.S. here. Unfortunately, it was accidentally introduced to Southern California, and it's been causing big problems there with the grape industry, over $100 million impact yearly. It's got a broad host range like most of the leafhoppers, and it's a highly effective vector of Xylella, so we were assuming that this would be one of the main culprits that we'd be working with. If you're not familiar with muscadines, they're delicious and highly nutritious, and there's a lot of potential for this as a growing industry in the southeastern U.S. They're native here, and as a result, they're well adapted to our climate of high temperatures and high humidity, which some of the other grapes aren't as able to withstand. And there's a thought that they have larger vascular cells that are less susceptible to freeze injury, which allows them to do well in our climate here in the southeast. And these larger vascular cells may also help to prevent bacterial clogging as well. And as a result, muscadines are somewhat tolerant, maybe not quite resistant. We're not exactly sure of the mechanism here, but they're said to be, certain cultivars are said to be tolerant of Pierce's disease when the vines are healthy. So if a vineyard manager is using good cultural practices and keeping the vines healthy, they can do quite well with very few chemical inputs. But under the warming climate scenario that we're heading into, heat stress is something that can predispose vines to other injury and make them more susceptible to diseases like Xylella. So the objectives of our research here were to determine the species diversity and abundance of the leafhopper populations in a couple of experimental vineyards in South Mississippi, to determine the seasonality of these species and their distribution amongst 5 cultivars. We also wanted to take an assessment of the Xylella infection within the vines and ultimately try to sequence the Xylella DNA from captured insect and plant samples. To do this, we used 5 Muscadine cultivars, and 4 plants per cultivar were tested at 2 different research vineyards using yellow sticky cards. The traps were changed out bi-weekly from April through October over 2 years. And in addition to these trap counts, I also took fresh leafhopper specimens directly from the cards and preserved them in ethanol to facilitate our qPCR analysis to determine if we could identify some Xylella within the insect tissues. And we also collected leaf samples from each cultivar. You can see here that in the first year, the glassy wing sharpshooter was indeed our predominant species. Nearly half of our insect specimens collected were glassy wings. But in the second year, we had something interesting happen. This species, Siphonia orientalis, was something new to us. It's an invasive species, and it really took off in the second year. It became the predominant species, and so cause for concern perhaps. The site breakdowns, there were some interesting differences among our 2 sites. This site in Pearl River County had significantly higher populations of glassy wing, Siphonia, and Graphicephala virsuta compared to the other location. The most interesting thing here to me was in the second year, again, seeing that Siphonia population jump up really high, particularly at this McNeil site. The seasonality, you can see for all 3 of these primary species, there's a peak that begins in mid-May, and that's a potential recommendation for chemical applications if they're deemed necessary by a vineyard manager. Again, in the second year, you can see a similar peak mid-May forming, but with the Siphonia, we saw the early peak and then a second peak later in late July, early August. And in the second year, that big jump happened late. And if we can determine through our PCR analysis that Siphonia is a vector, this could be another potential treatment recommendation of late summer. With the cultivars, you could see here with our glassy wing sharpshooter counts, the Southern Home cultivar in both seasons was a preferred cultivar for the glassy wing. And with the other species, it wasn't quite as much notable differences. So in conclusion, the Southern Home, it was interesting to see the preference there. This cultivar has a pedigree that's shared between Vitis vinifera and the Muscadine rotundifolia. And so there's some interesting research to look into for the future there. The growth pattern is quite a lot bushier. There's more vegetative tissue on this cultivar, and so it may just be a facet of the microclimate, a higher humidity in there, and just more food, more vegetative tissues to support a leafhopper population. But that'll be something interesting to look at going forward. Collected over 30 leafhopper species, but it was really those 3 primaries, the glassy wing, the Siphonia, and the Graphicephala virsuta were the 3 primary species, with peak captures from May into August, and a potential treatment recommendation of mid-May. Potentially another one for Siphonia if we can identify significant levels of Xylella in the insect tissue there. Unfortunately, our PCR analysis is incomplete at this point. We were able to identify the Xylella DNA from our glassy wing sharpshooter samples, but the other ones were inconclusive, and we need to take another stab at it here this summer. So please stay tuned. Thank you.